Then we will talk about real-time PCR primer design. As you remember, in the cells, we will always have the chromosome DNA, and then our cell will start generating messenger RNA using the genomic DNA as template. However, in the genomic DNA, you contain the intron, not only the exon. But for the mature mRNA, the intron of this part will be excised, will be removed from the messenger RNA, become a mature messenger RNA. And then when we extract RNA from the cells, we will only extract the mature ones and then do the reverse transcription, which means that your cDNA, also your mature mRNA and the cDNA synthesized from it, will contain no introns inside, compared to we have the intron in the chromosome DNA. Even though you have performed DNA digestion during the RNA extraction, there is still a chance there might be some genomic DNA left in the sample. And since in the gene expression analysis, we are actually want to detect the amount of messenger RNA for the sample, not the genomic DNA. We therefore do not wish to amplify any products from genomic DNA to cause false positive phenomenon. So it is crucial to design a set of primers that won't amplify products from genomic DNA. Imagine we have 4 scenarios, and let's discuss what are the pros and the cons for these strategies. The first one, if you design primers within exons, then you will amplify the region of the exon 2 in this case, but this primer set will also be able to anneal to the chromosome as well, because the exon 2 exists in your cDNA, also exists in the chromosome. The next one is the intron spanning methods. Imagine that you have the forward primer on one of the exon 1, and then you have the reverse primer on another exon 2. And then you can use these primers to amplify PCR products from your messenger RNA or from your cDNA. However, these primers also will anneal to the chromosome as well, and then will amplify longer PCR products from chromosome DNA, since it amplifies intron that is not exist in cDNA but exists in chromosome DNA. The above 2 methods are not ideal. And the third one is exon spanning. Imagine that you design a primer, a forward primer on exon 1, and then you decide to design a reverse primer on exon 3. For the messenger RNA or cDNA, we will amplify the region from exon 1 to exon 3, this segment. However, for the chromosome DNA, these primers also can anneal on exon 1 region, and the exon 3 region correspond to the genomic DNA. Therefore, if genomic DNA contamination exists, primer will anneal on it and start amplify the region of exon 1, 2, and 3, and another 2 introns in between. So you will get even longer PCR products. That's not ideal for you as well. The best way is to design the primers to have at least partial region on exon 1, and then this forward primer will have some nucleotides designed on exon 2. So you cross exon-exon junctions. Same thing applies to reverse primer. Partial of the oligos will design on exon 3, and the rest of the oligos of reverse primer will design on exon 2. In this case, the messenger RNA will be used and the cDNA will be used, that is without intron. For them, the exon junction, the primers designed on exon junction will help let the primers only anneal to the exon 1 and exon 2 junction, and exon 2 and exon 3 junction for forward and reverse primers respectively. But these primers will not be able to bind to the corresponding segments on the chromosome DNA, because remember that there are several oligos exist on exon 2 and the exon 1. So chromosome DNA, they have the intron in between. Therefore, the primer cannot anneal properly to the chromosome DNA. So that will be ideal. But sometimes we don't have this ideal scenario. So at least you can make sure if the messenger RNA or if the gene you are going to amplify have at least one intron, you can design your forward primer between the exon-exon junction to cross the intron region. So now you know it is better to design primers that cross exon-exon junctions. Here I'm going to show you how to do the design using a gene with known mRNA and corresponding genomic DNA sequence.